So here's your babies in 4D, and as we can see, they're identical.
Identical twins are as close as nature can get to human cloning. Tiny replicas with identical genes, wonders of human reproduction. Some are amazing mirror images, their movements perfectly in sync. Others can be separated for years, only to discover they share strikingly similar jobs, tastes, and hobbies. But not all identical twins are as identical as we once thought. Advances in genetic science are revealing key differences in personality, development, disease, even sexuality. Through state-of-the-art 4D ultrasound scans, scientifically accurate visual effects, and microscopy footage, we delve into the complex yet delicate world of twins in the womb. In a world where most of us look different from one another, identical twins break the mold. But it's not their similarities that prove most fascinating to scientists. It's their differences. These boys are identical twins, but born with dramatically different birth weights, they hardly look related. Twins like these, with major physiological differences, present an enormous opportunity to reveal truths about human identity. From fingerprints and intelligence, disease, and even sexual orientation. Identical twins begin life like everyone else, as a single cell, a fertilized egg called a zygote. This cell contains 23 pairs of chromosomes, tightly packed strings of genes. One set is from the mother's egg, the other from the father's sperm. Together, they create the entire genetic blueprint for a new human life. A few hours after fertilization, the cell, with its cargo of chromosomes, embarks on a remarkable journey. It travels along the fallopian tube toward the womb. Then it starts to divide. First in 2, then 4, then 8, each cell an identical replica of the other. This rapidly multiplying collection of cells is called a blastocyst. And it's here that nature's most remarkable reproductive anomaly can sometimes occur. It's an event so mysterious, it wasn't witnessed until recently. Several days after conception, the blastocyst spontaneously splits in two. Each new blastocyst is composed of cells with identical sets of chromosomes, and they carry the same arrangement of genes along their length. The two blastocysts now have the potential to develop into identical twins. Exactly how and why the blastocyst splits is one of reproductive science's greatest mysteries. And it wasn't until 2007, during a lab study of 26 embryos, that embryologists witnessed this event for the first time. The blastocyst's outer membrane is called the zona pellucida. The lining inside is called the trophoblast. Just one cell layer thick, the trophoblast will develop into the placenta. The inner cell mass, filled with embryonic stem cells, will become the fetus. Over the course of 4 days, embryologists noticed that a junction between cells in the trophoblast membrane would regularly weaken, letting liquid leak out and causing the trophoblast to collapse like a balloon full of water. Then it would repair itself. But in 2 of the eggs, they saw something for the very first time. After the membrane collapsed, the inner cell mass divided in 2, each clump sticking to a different side of the trophoblast as it reinflated. If these clumps were to develop in utero, they would grow into identical twins. Born from the same single fertilized egg, it's not surprising that most identicals retain a similar physical appearance as they grow up. But genes influence far more than our looks. They affect personality and taste, making us flamboyant rather than shy, with a taste for coffee instead of tea, and a love of routine versus a desire for experimentation. Similarities between twins are often more than skin deep. Dennis and David Herrera are 53-year-old identical twins from California. After high school, both became officers for the Los Angeles Police Department. They were raised together, went to the same school, and had the same friends. But it's impossible to say whether their career choice was influenced by their genes or their upbringing. To test the extent that our genes influence our life choices, scientists study identicals separated at birth and raised in different environments. Daphne and Barbara were separated as babies and had no contact with each other for 40 years. One was raised by a scientist, the other by a municipal gardener. But the similarities between them are striking and probably go beyond coincidence. They both left school at 14. At 16, they both fell in love. In their early 20s, they each married their childhood sweethearts. They both suffered miscarriages at the same age, but went on to have children in their early 20s. Both have an irrational fear of blood and falling down. When they met for the first time on a train platform, they wore almost identical clothing. The similarities suggest that genes play a powerful role in shaping many aspects of our lives and personalities. But it's the differences between identicals that could offer science the greatest opportunity. The chance to solve the age-old debate of nature versus nurture. When these twins were born, Jake weighed a healthy 5.5 pounds. His twin Tom weighed just over 1.5 pounds. Since they're identical twins, this dramatic difference was a mystery. So, doctors looked to the one environment the boys shared before birth for answers: the womb. By day 5, the two identical blastocysts reached the womb. Now they face one of the most critical events in their 9-month odyssey, one that could dramatically affect their lives. In the womb, and beyond. When they were born, Jake weighed 5.5 pounds, his brother Tom a mere 1.5 pounds. Yet they're identical twins, born just minutes apart. How can twins with the same genes look so different? The answer may not be in their DNA, but in events that occurred in the environment they shared before birth, the womb. The first, most critical event is implantation. Around day 6, the blastocyst must implant in the wall of the womb to secure its position for the next 250 days of growth. Timing is everything. Each twin will rely on a placenta to supply it with oxygen and nutrients, but the placenta only starts to develop after implantation. So blastocysts that split early before they implant each get their own placenta, while those that split late after implantation have to share one. A shared placenta may mean that one twin receives less nourishment than the other. As a result, it grows much slower, a condition called selective intrauterine growth restriction. There are even rare instances when identical twins have their own placentas and still develop this condition. Jake and Tom are one such case. Their different weights are the result of events that occurred long before their birth, but the effects may last a lifetime. Once implantation occurs, embryonic development is surprisingly rapid. By the end of the 3rd week, a rudimentary body shape is already emerging. The embryo resembles the beginnings of a body, totally unprotected by skin and bone. On one edge, stem cells flow over and are assigned their fate by the organizer. One of the first organs to form is the heart, just a clump of muscle cells until now. Around day 22, a single cell in this dormant clump spontaneously contracts. It sets off a chain reaction. One after the other, neighboring cells start to contract until the entire vessel pulsates. Two tiny hearts, about the size of 2 pinheads, beat together. The twin embryos have approximately the same number of cells and the same DNA, but they may already be different. And those differences will become deeper and more profound as they develop, shedding light on some of the deepest mysteries of human identity. Is personality innate or learned over time? Why do some of us succumb to disease while others remain immune? Where do we get our skin color? What makes one person gay, another straight? the answers aren't as simple as we once thought. Remy and Kian are fraternal twins, the result of two separate sperm fertilizing two separate eggs. The girl's parents are of mixed ancestry, which usually means the children will resemble them, but not always. Those with mixed ancestry carry a mixed set of genes for both lighter and darker skin. In Remy's case, a sperm with a set of genes that produce darker-skinned children fertilized an egg with similar characteristics. In Kian's case, a sperm with genes that create lighter-skinned children also fertilized a similar egg. The result? Twins, but twins who look very different. Remy and Kian are living proof that twins can often confound expectation, but they aren't the only ones. In 2001, a pair of twins was born who appeared to be a boy and a girl. On closer inspection, it turned out that one had signs of both male and female genitalia. The twin was a hermaphrodite. Scientists believe this happened because a single egg was fertilized by two sperm, one with a female sex chromosome and one with a male sex chromosome. The egg developed into a blastocyst with both male and female cells. Then the egg divided, and each of the two new cells shed an extra chromosome, resulting in a blastocyst composed of both male and female cells. When the blastocyst split to create twins, one had more cells with the male sex chromosome and developed into a boy. The other had cells with similar numbers of male and female sex chromosomes, resulting in a child with male and female genitalia. The twins are halfway between identical and fraternal, the only known case of semi-identicals in the world. Identicals, fraternals, and semi-identicals. The simple classifications we're used to may be falling apart, but scientists continue to close in on the genetic riddle of why identical twins occur. At 5 weeks, the identical twin embryos are starting to take shape. Curved into a C, their heads and tails can be distinguished, along with their hearts, spinal columns, and tiny limb buds. At this stage, the embryos look almost indistinguishable from any vertebrate in the animal kingdom. Yet even at just 1.5 millimeters long, these fragile creatures are about to embark on a critical period of brain development. This 5-week milestone could be another stage where identical twins develop key differences before they're even born. Celso and Jesus Cardenas are identical twins who were raised together. They remained physically similar as they grew up, but their tastes and interests began to diverge. Celso became interested in dance and academia, while Jesus preferred sports. The most surprising difference between the two brothers is that Celso, wearing black, is gay. The differing sexual orientation of identical twins allows us to investigate one of science's most controversial questions. Are people born gay? Celso and Jesus were raised by the same parents in the same household, so they shared the same environment at a crucial time in their personal development. In the general population, the chance of someone being gay is less than 5%, unless you have a gay twin. Then the chances are much higher. If you are fraternal, sharing half your genes, there's nearly a 25% chance that you will also be gay. If you are identical, sharing all your genes, there's about a 50% chance you will also be gay. This suggests that there must be some genetic component to our sexuality. But it can't all be up to genes. Otherwise, every identical pair would either be both gay or both straight. Some other factor must be at play. In their first few weeks, all fetuses develop along similar lines. If nothing changed, each one of us would be born female. Fetuses with the male Y chromosome will form testes at about week 6 that produce the hormone testosterone. But at about the 8th week, the testosterone is released and may affect early brain development. Testosterone masculinizes the body. It also masculinizes the brain, including the hypothalamus, which partially controls who we find sexually attractive. Some scientists believe that the more the hypothalamus is exposed to testosterone, the more it sets the stage for a sexual inclination toward women. Occasionally, a male fetus doesn't produce sufficient testosterone, or its brain doesn't absorb enough to shape it along heterosexual lines. If this theory is correct, then it may be that Celso absorbed enough testosterone to masculinize his body, but not enough to fully differentiate his brain. As a result, he was left with a desire for men. Many mysteries remain, but twins like Celso and Jesus play a crucial role in informing scientists about how and when sexuality develops. Week 8, and the identical twins have finished their embryonic stage of development. They are now known as fetuses, meaning offspring in Latin. The twins are secure in their own sacs, but share a placenta. They have approximately 200 days left until birth. This is when their growth accelerates. At 13 weeks, they've started to look more human. Their eyes have moved together, and their heads are in better proportion to the rest of their bodies. At the end of the first trimester, most pregnant women will have their first ultrasound. The ultrasound sonographer checks for basic signs of normal development.
So here's your babies in 4D, and as we can see, they're identical.
The sonographer sees a single placenta, a sure sign that the twins are identical.
We can see here a single placenta, like a T-section, which proves that you're having identical twins.
So when would you see them start interacting or reacting against each other?
We can see them almost sort of moving.
From now on, the mother will be closely monitored. Twin pregnancies carry a higher risk of premature births. Immature development of lungs and organs can lead to complications. These complications also make natural childbirth more dangerous. So, twins are about 50% more likely to be born by cesarean section than single babies. It's now the start of the second trimester, 12 to 24 weeks. During this period, the twins will experience not only dramatic growth, but massive development in everything from the structure of their brains to the refinement of their facial features. By the end of this stage, the fetuses will be small, but almost perfectly formed. They look very similar, but new research reveals that even at this early stage, identical twins may already be less identical. Subtle changes are underway that will become greater throughout their lives. And the mechanism responsible is one of the most complex, fascinating processes of human development. "15 weeks into their journey from conception to birth, the identical twins move for the first time. They interact and explore their environment with their hands and feet, touching and even appearing to kiss. Eventually, their mother will feel the same kicks each twin feels in utero. Sometimes they appear aggressive, other times almost caring. They look identical, but unseen influences may already be taking effect, ones that could subtly alter the expression of genes in their growing bodies. Identical twins Jake and Tom are now 17 months old. Jake is 3 inches taller than Tom and nearly 9 pounds heavier. When they were born, doctors attributed these discrepancies in size and weight to restricted nutrition or blood flow in the womb. They expected Tom to catch up. But after a year and a half, they're investigating whether Tom is suffering from a genetic condition called Russell-Silver syndrome, a growth disorder. One of the causes of this syndrome may be a problem with a growth gene on chromosome 11. Scientists are puzzled. Tom and his brother don't have the same syndrome, but identical twins have identical genes. So, shouldn't they? Genetic puzzles like these are opening up the outer reaches of science, called epigenetics. This growing field of biology may help explain the differences between twins like Tom and Jake. Epigenetics reveals that even if their DNA code is the same, the way it functions can differ. The human genome contains around 25,000 genes, each with its own specific function, like producing energy or directing cell division. Now, geneticists are investigating a previously unknown aspect of the genome called the epigenome. A series of chemicals that act like switches are capable of activating or deactivating individual genes. One of these switches works by a process called DNA methylation. Enzymes inside a cell attach a minuscule molecular compound, a methyl group, to a gene. This compound can deactivate or at times activate the gene, but the gene itself remains. The cell's DNA profile is unchanged. This could explain the difference between identical twins, Jake and Tom. Some unidentified environmental factor in early cell development caused a methyl group to attach itself to a gene on one of Tom's chromosomes, a gene associated with growth. The growth gene is still there, but it's been switched off. By studying twins like Tom and Jake, geneticists are finally finding clues to some of the great mysteries of the human genome. This is a map of chromosomes belonging to identical twins from age 3 on the left to twins aged 50 on the right. The red markings are an indication of the increase in the methylation that has occurred to their chromosomes over half a century. Factors like smoking, diet, and chemical exposure can wreak havoc on genes and affect how they operate. And there are different environmental influences not only in our lifetime, but also in utero. Hormones, space, and quality of nutrition can all affect identical embryos differently, even those sharing a placenta. The activation and deactivation of genes during early development could explain many twists of fate that affect us all. Why one person is struck by disease and another is spared. Epigenetics may also play a significant role in determining sexuality. If sexual preference is associated with an unidentified gene, it may be that the epigenetic suppression or activation of this gene dictates sexual preference. These genetic switches may be the answer to why one twin absorbs more testosterone than the other, resulting in one being gay and the other straight. It's becoming clear that our health, personality, tastes, and even appearance aren't the product of our genes or our environment, but that nature and nurture are inextricably bound, with epigenetics the biological link between the two. We may even have the power to steer our genetic destiny and that of our descendants through lifestyle choices like smoking, diet, and exercise. What makes identicals unique and useful to genetics is that their genome is a 100% match. Or at least, that's what we thought. Now, powerful new DNA techniques are revealing a different story, one that is only just unfolding. As the second trimester draws to a close, the twins have a second scan. The development of external features like limbs and eyes will be checked, as well as the major organs, for signs of defects. The twins are about 8 inches long, small enough to cradle in the palm of the hand. The 20-week scan is when most mothers discover whether they're having a boy or a girl. Gender identification can be done as early as 11 to 12 weeks, but is more definite at 4 months. The scan reveals that these twins are boys. The twins are 24 weeks into development. Their mother can now feel them kick. A battle for space rages inside the womb. A scan reveals this struggle and maybe even a glimpse of emerging personalities. Ultrasound studies, combined with observations of twins after birth, show very early forms of shy or extrovert behavior. In one study of fraternal twins, the shyer twin would occasionally hang on to his umbilical cord and even appear to lick it, while his sister would grab, kick, and push him. This pattern continued after birth. And at 4 years old, the shy twin still exhibited reserved behavior, while his extroverted sister would grab and play with him. Our personalities may start to form in the womb after all. From week 29 on, the fetuses finally resemble newborn babies. Their body fat rapidly increases. Bones are fully developed, but still soft and pliable. Head hair grows thicker, and fingernails reach the fingertips. The twins' fingerprints are already different. Epigenetic changes have impacted the genes that control the patterns, and the differences will become more pronounced. As the fingertips develop, they receive blood from the veins and swell, forming distinctive fingerprint patterns. If blood flow from the placenta is restricted in one twin, blood may be directed away from the fingers and other extremities, leaving it with more fingerprint whorls than the other. Our fingerprint patterns can help indicate how healthy our heart will be in adult life. The more whorls, the greater the chance of heart problems. From the moment a fertilized egg divides in two, various influences on the genetic code conspire to create differences between identical twins. Differences that can become more pronounced over time. But it's always been assumed that identical twins share identical DNA. Until 2007, when a discovery surprised the scientific world. Researchers set out to investigate one of the most puzzling anomalies in the life of twins: why one succumbs to a genetically inherited disease while the other is unaffected. They chose Parkinson's because it's more likely to be caused by a spontaneous genetic cause than inherited. The scientists examined the genes of 19 pairs of identical twins, including 9 pairs in which only one twin showed signs of the genetic disorder. The goal? To challenge the common perception that identicals are genetically identical. Their analyses showed differences between the numbers of genes within each twin. Genes usually occur in 2 copies, one inherited from each parent. But this study revealed it's possible to have just 1 copy, or have 3 or more copies of the same gene. This phenomenon is called copy number variation and can fundamentally alter the gene's function, inducing disease in one twin and not the other. It's a major breakthrough in our understanding of disease and how it affects twins. The discovery means that identicals are not a 100% genetic match. The very definition of identical twins needs to be revised, as advances in technology prove that small but significant changes are present in all identicals. The identical twins are approaching full term, already with subtle genetic differences between them that may shape very different futures. But first, they must face the most dangerous stage of their long journey in the womb: birth. It's 35 weeks. The twins are now fully developed. They have hair, eyelashes, nails, and can open and close their eyes and mouths. Conditions in the womb are cramped, even though at 4.5 pounds, each weighs a third less than the average baby. But combined, they put tremendous pressure on the womb. Premature deliveries for twins before full term, at 37 weeks, are common. Our twins are ready to be born at week 35 by C-section. About half of twins are delivered this way, due to the difficulties and dangers of giving birth to two babies naturally. There's a larger than normal delivery team on hand to ensure safety. The obstetrician cuts the first membrane, then the second. Cesareans are always performed quickly to reduce the risk of complications. The doctor plunges his hand into the womb and takes the first baby out. The baby takes his first breath and emits a healthy scream. The cord to the placenta, his lifeline for the last 35 weeks, is finally cut. Now it's the other twin's turn. With his brother out of the way, he follows quickly, also head first. It's not always obvious to the obstetrician whether the twins are identical or fraternal. But our twins shared a placenta, the clearest indication that they're identical. If the identicals have separate placentas, which happens occasionally, DNA analysis is the only sure method of identification. After an extraordinary journey, our two reproductive marvels have made it safely into the world. They may look alike, but we know they're already different. "245 days ago, a fertilized egg divided, creating two identical embryos. From that moment on, their paths began to diverge. Inequalities in nutrition or space, different exposure to hormones, small alterations in their epigenetic profile, even minute differences in the underlying genome, all combine to create subtle yet significant differences. Now, the twins are about to embark on the next stage of their journey. As they grow, the differences between them will become even greater as they face diverse experiences and influences. These differences bring us closer to answering some of humanity's most profound questions. The study of identical twins has shed new light on the complex mix of genetics and environment, nature and nurture that makes each of us unique. It has profound and far-reaching implications for how we all lead our lives.